First, I would like to say that I gave the very first talk of this conference last week, on Friday. And it has been a wonderful week, a long week, but I learned so much, and I enjoyed very much the city. So I'd like to thank one more time the organizers, and I would like to thank the organizers here in Sofia, because it has been a great meeting. So in my lecture last week, I focused more on the inference methods that we use to assess genome architectures, mainly mammalian. Okay, so this is the pipeline that you have seen several times already in this meeting. We start with epigenetic markers, we go through machine learning to define these chromatin structural types, and then we use physical simulations to predict an ensemble of 3D structures, which can then be studied and validated. So the general idea behind this pipeline is that the chromosomal ensembles are determined by sequences, just like the protein ensembles. In the protein ensembles, you have a sequence of amino acids that determine the structure of the protein. In chromosomes, you have a sequence of chromatin structural types, which are mainly determined by epigenetic information. And this sequence of chromatin structural types determine the structural ensemble of chromosomes. The crucial difference is that while the sequence of amino acids is static, the sequence of structural types can be dynamically rewritten to accommodate different folds for the same DNA. So, in terms of what are the molecular mechanisms that shape the structural ensembles of chromosomes, a few years ago, together with my collaborators, we hypothesized that there were two main drivers. The first is the phase separation of chromatin structural types. So you have that when two segments of chromatin come into contact, the energy of that contact depends only on the type identity of that contact. And this was sort of an inference from high-C data, which you can see that the patterns of interaction, they line up very well with epigenetic information. Here, when you see this pattern, for example, you can always see that there's this H4K20 trimethylation is present. Similarly, you can see that in these two human cell lines, there's changes in epigenetics that result in changes in the structure for the same DNA. This is human DNA, so it's same identical DNA. So this was the thinking behind the first hypothesis, which was phase separation of epigenetically driven phase separation. And the second hypothesis that we made is this that we call the ideal chromosome. And when you look at the structural ensembles of chromosomes as they arise from experiments, you can see that there are very specific statistical properties for the polymer, and they come from the high-C maps. So we thought, okay, let's introduce an energy term that depends on the genomic distance. That would, kind of help us study the statistical properties. Another way to see that is to see this as a process of discovery. For example, here you see Linus Pauling, and you have I+4 interactions, which are the ones generating alpha helices in proteins. So this is a structural motif that it exists and is developed by evolution to create the fold of proteins, but that can be discovered by learning the energy terms from high-Z maps in this case. So later on, the process of DNA extrusion was discovered, and it became clear that the ideal chromosome, as we were fitting from high-Z maps, was indeed due to motorization. So, and this is sort of the slide. So here you have the process of extrusion. But it's also important to notice that by fitting the high-Z maps, yes, we're fitting some of the activity of cohesin and SMC complexes, but we might be fitting also the activity of other motors that haven't been discovered yet. So these two hypotheses over the molecular processes shaping chromatin organization lead to this energy function. And this energy function includes a polymer potential, includes these type-to-type interactions that are determined by epigenetic information. They generate phase separation, and they act exclusively across Cartesian space. And then you have this other term, which is the ideal chromosome term, which includes interaction along the polymer fiber. All these models that we mentioned, they walk along the fiber, and therefore they carry implicit memory of a genetic distance. So this term depends both from Cartesian distance and genomic distance. So then, as I discussed in my previous lecture, you can fit the parameters in this energy function from high C, just like in the same way you fit an empirical energy function for protein folding from crystal structures. And then we go back to these slides which I've seen multiple times. We have discussed how we do the physical simulations. And now there's the part of doing the inference from epigenetics to chromatin structural types, which Ryan has discussed, and this was work we did together a few years ago. What we do is we read the epigenetic sequences and we predict what is the best, what is the most probable chromatin structural type, given the epigenetic distance, the epigenetic information which we can get from ENCODE database, for example. And this is done with a statistical model which resembles a POTS model or a half-field neural network or an Ising model, depending on which field you're coming from. So anyway, this was completing the pipeline. Now we have an ensemble of structures. When I say an ensemble, I mean 50,000, 100,000, 200,000 structures for this single chromosome. And we can go compute in silico high C. So we take our structures, we compute the high C, and then we go compare it with the high C that's been determined experimentally. These are the famous GM12878 maps from Lieberman-Hayden lab, and they're very well predicted, almost at the same level of biological replicates. So we were very happy with that. One thing that at the beginning was a question, and I think it's less and less a question these days, is how much structural heterogeneity there are in these chromosomes. One could think there is a single native structure and then there's fluctuations around that native structures, or one could think it's a more fluid, liquid system which doesn't really have a single native structure. And our model predicted that the structures are fairly fluid and that there is microphase separation, and that microphase separation is a dynamical process with this liquid droplet splitting and fusing in time. This was back in 2016, 2017. We paid for a graphical artist to represent these ideas in this picture, which is now very similar to the picture you see arising from the field of condensates and phase separation and liquid-liquid phase separation. So this was coming out of our model. Now the question is it right? Is it wrong? So we started looking at validation from fish, which can sort of has the information about individual cells. And we could reproduce the profile with fish. This was a 3-probe fish done by Lieberman-Hayden in 2009. So it was old data, but we could predict it very well. And this sort of like is carrying the signature of phase separation. And we could predict that, and we could predict the spread of this distribution, and we could predict the distances, which is remarkable because the model was trained on high C, and high C doesn't know about distances. Here we were predicting distances from ChIP-seq information. So that was a very reassuring result, even though the data they were not particularly informative because it's old data. Then came DNA tracing, and all of a sudden we could do a lot more. So Ryan Cheng has done most of this work. We could look at the G of R of the predicted ensembles and of the imaged ensembles, and they overlap very well. We could look at the heterogeneity. We could do clustering. And it's important to notice that this imaging came after the predictions. So we didn't fit this data. This data were predicted a couple of years before. So this was good. That gave us confidence, and we started applying the same approach to different human cells and see if we could predict different cell lines directly from epigenetic information. So how does the epigenetic information of different cell lines determine the structures of different cell lines? And that worked very well. As usual, you have the experiment on bottom and the simulation on top. You can see, for example, that this epithelial cell, they have this big block here, which you find here. The lymphoblastoid don't have that block, and in fact, we didn't predict that, and so on. So you can read the paper. This is a paper that was published 2020. And this line of research sort of like now is to the engineering stage. All the colleagues at CTBP, they're now working together with the Aiden Lab and in collaboration with the NIH ENCODE project to predict structural ensembles for a variety of human cells and tissues. You can already go online and get the files for all the structural ensembles of these cells. So at this point, we're fairly confident that we have a good working pipeline. It's validated extensively. So now I'd like to go back and discuss a little bit what did we learn from these early attempts? What did we learn from the model? What did the inference from high-Z teach us about the structure of chromosomes? So we go back in time. Back in 2016, it was immediately evident that it's heterochromatic interaction that are driving the compartmentalization, that are driving the structure of chromosomes from the point of view of phase separation. And that came out because when you flow fit the energies of the model to the high-Z, then you find that this B-B interaction, B is the heterochromatic compartments, they're much stronger. So this we noted immediately. It was a result of the statistical inference, and physically speaking, what that means is that when you have chromosomal territories, you have that the active chromatin is positioned at the periphery of territories. So we say that in 2016, we repeated that in 2020, and then in 2020 comes out DNA tracing showing that this map of transchromosomal proximity frequency matrix, which you see that the A compartments are the one that constitute most of the interchromosomal contacts. So that was a direct confirmation of our prediction from imaging standpoint. Then we go back. What about this ideal chromosome term? Well, once you fit it, the ideal chromosome turns out to be lengthwise compaction. That's what the data say. And what we found immediately is that this lengthwise compaction had a big influence in the formation of chromosomal territories. This is a figure from our paper 2016, in which we immediately confirmed that we could get the territories because we knew that this was an important feature of chromosomal organization. And we wanted to be sure that we were able to model that. So, and this term of lengthwise compaction is also intriguingly very important in keeping chromatin unknotted. So what we did here, I think José just showed this slide already. What we did here is we started the simulation with a homopolymer, and a homopolymer is characterized by a distribution of entanglement values, which is this blue distribution. Then it's fairly knotted. And what you do is we turn on the lengthwise compaction, and this distribution, this blue distribution, which is the knotted distribution, move to the red distribution, which is fairly unknotted. Of course, when we say knotted and we say not knotted, it's still from the ensemble standpoint. This is 100,000 structures. There's going to be some knots, but it's clear that the lengthwise compacted polymers have a lot less knots. So this was interesting, and that sort of made us rethink a little bit about the history of this of the problem of entanglement and chromatin topology. So in the center, you have sort of like a model, which is a very good model. This is early work from Grossberg. In this idea, you have the fractal globule, which is long-lived, non-equilibrium state of a collapsed homopolymer. So this polymer is unknotted because it's created without knots, and it cannot relax to equilibrium because you don't allow chain crossing. If you were to wait a very, very long time or allow chain crossing, for example, by topoisomerases, the equilibrium state of this model is the equilibrium globule, which is knotted, as we showed in the blue distribution. Now, the micron globule is different because it's an equilibrium state of a lengthwise compacted polymer. We do let the system equilibrate because we do let the chain cross each other because we do believe in the activity of topoisomerases. And this model basically removes the knots. Even if you started with a lot of knots, it goes down to its minimum free energy state, which is largely unknotted. So this was sort of like, kind of like a very, topologically, these two models are very similar, but the process is very different. And this was, we showed it in 2016, but then people started doing simulations with explicit extrusion, and sort of they confirmed this notion that it's really the extrusion and the motor activity that removes the knots in one case actively. In our case, sort of we have this energy function that there's a mean field energy function for this system, which is an equilibrium energy function. So these papers later on with active processes sort of show the same concept. So here we started thinking that this sort of like these polymers, these lengthwise compacted polymers, is sort of like a new class of polymers which are have very rich behavior, and we started looking in that direction. So this was sort of like our state of mind when we started going to looking how these molecular processes they act along evolution. So here's the figure that you saw from Olga Dudchenko. They've done these experiments. This is high C, 24 species. What high C tells us in this case is that there's a variety of different architectures along evolution, which can be mainly recapitulated by a type 1 architecture in which you have territories, and a type 2 architectures in which you have either telomere-telomere clustering or centromere-centromere clustering, or this sort of foldback of the structures. Now, the telomere-telomere clustering and the centromere-centromere clustering, they show up as this bright peak in the interchromosomal quantum maps. The foldback shows up as a secondary diagonal here. So, and these three features are sort of like very correlated among each other. When you have one, you tend to have the other two. So we call this type 1 architecture and this type 2 architecture. Then there was, and then Olga discovered that the type 2 architecture was very much correlated with the absence of some functional units of condensin 2. Somewhere around the same time in the Netherlands, Claire Owenkamp and Ben Roland, they were doing these experiments of knockout, which they were knocking out condensin 2, and they were seeing this this new phenotype arising, which would go from a territorialized high C map to a high C map which has clustering of centromers and telomers. So when I saw this data, I was like, I know what's going on. I mean, it's very clear that from our Hamiltonian, if I knock out condensin 2, which is an extruding complex, then I keep everything steady, but I need to decrease the ideal chromosome. I need to decrease the amount of lengthwise compaction, which is what we did. And all of a sudden, we go from a phenotype with a wild-type phenotype that has territories to a phenotype which has less lengthwise compaction, which has these sort of dots that show the existence of the clustering of centromeres and telomers. Okay, so I'm gonna hurry up. So Sumitav Brahmachari, who was the main person working on the theory on the science paper, pushed this forward, looking at the sort of the phase diagram of what happened when you have short lengthwise compaction versus long lengthwise compaction. Of course, there's a bunch of models. Each model has its own propensities. And you can achieve a bunch of different structures just modulating the shape of the lengthwise compaction. You can go from globular to strings to rope-like. Then he included the presence of lamina. And you can start writing, a little diagram which you see what is in competition with what and what supports the other activities. And this was published in 2022 in Glycadis Research. And at the same time, Vinicius Contesolo, who's sitting there, was taking another approach to the same problem of understanding what are the molecular determinants of organization in different organisms. In this case, we use the experiments from Aedes aegypti, the forgotten pandemics that Olga has mentioned. So this is an organism that has a very different organization from mammalian. It has the clustering of centromeres and telomers, and it has a very thick diagonal with secondary diagonal. So what Vinicius did is he did the new optimization. So he derived a new energy function for this organism, but still based on the same molecular mechanism. And what we get is that despite the fact you cannot see the territories in the high-C maps, the territories, they do exist. There are non-overlapping regions of chromatin for each chromosome, but they are very elongated, very distorted. Another interesting thing is that this thick diagonal. This thick diagonal means that there's a lot of short-range lengthwise compaction, and that leads to very thick chromosomes, resembling a little bit of mitotic chromosome. And that also leads to some ordering in the chromosomes. They look liquid crystalline, these coils of coils. That's been seen, I think, from Dave Tirumalai, Mirny, and co-workers. They also have observed some similar ways. They have sort of postulated some similar mechanisms. In this case, it derives from the data. It can be seen when you do the Fourier transform of the structures that there are two typical frequencies, a big turn of about 8 megabases and a short turn of 6 megabases. So this is an idea that Peter Wallinus has been pushing for a number of years, that lengthwise compaction needs to lead to these coils of coils, because that's just the physics of that happening. José has talked a little bit about the sensitivity to mechanical cues, so I'm going to skip that. Take-home message: chromosomes are fluid, yet organized. The genome organization mainly results from phase separation and motor activity. Epigenetic control, genome architecture along cellular differentiation. And we're starting to see that the diversity of genomic structures along evolution can be explained by a modulation of these two molecular mechanisms, which are mainly in competition. All my collaborators, my group, all the people working at CTBP, and you've seen that we really work as a group, so we share work and we work together. Our collaborators in the Netherlands. The funding, of course, we're always hiring. And thank you very much.
Thank you very much, Michele. Very nice talk. Question?
Great talk, thank you. Just a naive question. You see, you built, the structures that you have based on high-C. It's, when you do a high-C, all the time, there's some errors. This is because of the very nature of the cross-linking which is going through formaldehyde. So now, the question I ask is, when you just make the modeling, right, you have some structures. Is it possible after that, by analyzing, let's say, several either organisms or cells, etc., to go back and to design eventually which are the spots which doesn't correspond on the, which are false spots. So in other words, is it possible to correct I mean the experimental data.
It's a great question. So we actually thought about that. So let me just specify a couple of things. So the energy function is trained on High-C, but it's a lot simpler than High-C, right? So a High-C map is many gigabytes of data. Our energy function is only bits of data. It's 28 parameters over instead of like gigabytes. That has been done once in 2015, and it's still working. So, and the reason why it works is because, as you say, there's errors in the data, but by sort of aggregating the data, we have a buffered, filtered version of that. Now, to come to the second part of your question, that would be a great thing. So we thought, okay, so this is the energy function that I have from this data, but where do these two things disagree? Can I go back and figure out, is this point where the two data sets disagree depending on an experimental error? Is it a new process that hasn't been discovered yet? We are starting to do that. I don't think we have strong result on that yet, but it's a great idea. We thought about that.
Thank you.
I have a question which is even more naive. So you're talking about the energy function, but it seems to me that this is not actually energy. This is some kind of objective function in terms of optimization problems. Because I would think from other fields of physics, we know that these knots probably lower the entropy because they pin the chromatin. Like in the charge density wave in electronic systems, disorder pins the crystal. So is that true, or am I missing something?
It is absolutely true. That is always a reason of misunderstanding. This is an information theoretic energy function. Strictly speaking, it's a free energy. It's defined essentially as the logarithm of the probability. It is not a true energy, and that is an essential difference with other approaches. That said, having the inference of the effective of the mean field free energy function from the probability has helped us pinpoint real physical mechanisms, even though it's still statistical. There's no real forces, right? There's no real dynamics. There's no dynamics here. It's not, the temperature is complicated, the pressure is complicated, so entropy is complicated. So you are totally right, and I'm glad you mentioned it because it's an important distinction.
Okay, one more question.
Yeah, thank you. So you clearly showed that you can predict the 3D structure out of the epigenetic marks, but I was wondering how many marks are really relevant? Because, I mean, you started with the ENCODE, which is like a lot of data.
So great question. So when Ryan and I did that in early 2016, what we thought, let's just use all the data we have. So we throw in all the data that were available for GM12878. And the chip-seq tracks for the GM12878 at that time, there were about 120, 130 different experiments. We throw them in, we cooked the machine learning, we got some results. Now we started understanding that we didn't really need 120, partially because they're all correlated with each other. So the question is, how many do you need to sort of like sample the diversity of that data set, the cross-correlation? In the first paper, we reduced it down, I think, we had a model at 120 tracks. We had a model for 10 or 12 tracks. And I think Esteban, who's sitting there, now has a model for 5 or 6 tracks. So these days, we're acting in coordination with the ENCODE project. So on the same cell lines, they're doing always the same chip-seq, and tissues the same. They're doing a standard set of chip-seq. Eret's lab is doing the High-C. Esteban is predicting the chromatin the compartment annotation based on the chip-seq that they do, and then they run the simulations, produce the structural ensembles, and everything is online. You can go convince yourself if they're worth your time or not. I mean, but they're there. It's all available. So bottom line, you can do a reasonable job, A and B, with 4 or 5.
The point is you don't need everything that's important. You don't need everything that's important for the interaction. You just need enough tracks to differentiate between the compartments, subcompartments, right? So basically, as long as you have enough tracks, and the feeling we have right now, it's something between 5 and 7
Yeah.
appears to be sufficient, as long as they're strong. So basically, so what we're trying to do is, if we can differentiate using only methylation data, it's great, because every ChIP-seq has methylation. We have to add other things that will require more sophisticated ChIP-seq. But I think we can do a reasonable job using just, for example, methylation data, and in order to separate. Doesn't mean there's not other proteins that are important, it just means enough to separate the compartments using that.
ADAC-seq could be useful. We didn't use it very much because we're sort of stuck with ChIP-seq. I think you can correlate very well ADAC-seq with a number of ChIP-seqs. Yeah.
Okay, let's thank again.
Just accept.
Michele.
Just accept.